might remember a WhatsApp survey that Amol Akhade did about when do we look at the regulatory approvals at the time of deciding to treat the patient or not. And I think this is a very important thing in view of the easy accessibility of the information. All patients would come to us with information about the package insert and who has approved the drug. So we have a very important topic, and chairperson is Tarini. You are going to be there, okay. And Senthil needs no introduction, and he is going to talk to us about FDA approvals, Holy Grail of Oncology. Much awaited lecture, Senthil.
Good morning, everybody. Thank you, organizers, though I'm part of the organizing group. I want to thank Purvish sir, Amish, Sahoo, and Ullas for thrusting this responsibility on me, because I'm very comfortable moderating because you don't know answers to any of the questions that you're asking. But then suddenly I have to ask some questions and also give answers. So that's where this became a little complicated for me. But then, as I went about preparing for this talk, I learned quite a lot. And I hope I will be able to impart a little bit of what I learned, at least, to you at the end of this half an hour. So I'm starting with some disclaimers. I'm not an expert in regulatory approvals or biostatistics. I have my biases. I consult and speak on behalf of the industry. I'm happy to be challenged. I'm happy to be corrected also. And then something that I've not put up here, which came up yesterday, I've not used ChatGPT to prepare for this talk. Yes. So this is the appraisal of FDA approvals for adult, it's not slit tumors, between 2017 and 2021. The reason why this article was titled as Has the Eagle Landed is because the whole story behind rapid approvals, priority reviews, fast track was because of what was put in as the Cancer Moonshot program. Okay, and the bald eagle is the national bird of the United States. So this went back to look at what the FDA has done 2017 to 2021 to see whether the approvals that were done had decent benefits. And how did they quantify decent benefits? They try to look at the ESMO MCBS scale and apply it to FDA approvals. So how did we fare? There were 146 approvals for advanced disease, 15 for early disease. The early disease approvals were quite good. All of them scored A or B, 15 out of 15. But when you come to the advanced, we've not done very well. Only a third of all approvals were of substantial benefit based on the ESMO scale. About half of them had intermediate, and about a third again had, or 1 in 5 had poor benefits. So we are okay, but we're not really doing well at all as far as our benefits of therapy based on FDA approvals are concerned. We're all interested in immuno-oncology. Less than half of all immuno-oncology approvals were classified as substantial when you apply the ESMO's MCBS scale. So we are getting drugs. Are we getting great drugs? The answer is probably no. We need to do better. And the same article also looked at trends in approval pathways and disease settings, right? So the one on the top is the approval pathway and disease setting 1. So as you would expect, there are lower number of trials that are being done in the early disease setting. There are more done in the advanced disease setting. But what's important is the lower one. Trends in primary endpoints in studies. And what I want to highlight here is that a significant proportion of these trials were done with either response rate or pathological complete response as primary endpoints. And drugs were approved based on these endpoints. And even more important is the fact that 35% of all of these have been left hanging. We just don't know what has happened after that. We haven't gone back to look at these approvals. We haven't had confirmatory studies. Okay? Not a great picture. Now, how have our drugs, performed? Let's look at it in terms of absolute numbers. Just comparing immuno-oncology with genomic-targeted drugs, the incremental benefits that you get with genomic-targeted drugs seems to be better than what you get with immuno-oncology drugs. The median delta for overall survival, if overall survival was the primary endpoint in that study, was almost 5 months, which is not bad. And the range is anywhere from 1.5 to 49 months. It's a huge range. When it comes to immuno-oncology drugs, they've looked at putting all together and then taking lung cancer out of this. When you put lung cancer along with all of this, it's about 3.5 months. When you take lung cancer out of it, it's 2.5 months. So that's the delta for overall survival for immuno-oncology drugs. It's inferior compared to what you get with the genomically targeted medications. And how many of them have reported better quality of life? 1 in 5 studies. Most studies don't look at it. If you've looked at it, it's 1 in 5 studies which have really said that the quality of life is better for the intervention that you're looking at compared to what is standard of care. So, there's a trend in the right direction, and what this trend means is that compared to the previous decade, the delta is a little better. There are more studies that are reporting quality of life, but then you can imagine the way the previous decade was, right? It probably is terrible. If you call this dismal, that is terrible. But we're getting better. Let's not be nihilistic. Let's not be pessimistic. We are going in the right direction because the incremental benefits are getting better and more studies are reporting quality of life. So that's the report card. Now let's ask the question, what does the FDA do and is not doing, or what is not supposed to do? Let me read this. The FDA is responsible for protecting public health by ensuring safety, efficacy, and security of human and veterinary drugs, biological products, medical devices, and by ensuring the safety of our refers to USA, nation's food supply, cosmetics, and products that emit radiation. This is what is in the purview of the FDA. Now, how does it decide whether a drug should get approval or not? It is purely based on the benefit versus risk evaluation. Okay? It's purely based on the benefit versus risk evaluation. What it does not do, it does not have any binding on the cost of medications. Okay? The FDA has got nothing to do with cost. And it does not always consider how valuable or meaningful the benefit can be. Right? So it's supposed to, what it does is it tells you that the risk versus benefit is in favor of the benefit. So I am approving this. I don't have anything to do with the cost of this drug. And I'm not always going to look at value, benefit, and all of those things. I'm going to say, okay, this is what the statistical plan was. The statistical plan was, I have to show an X percentage in terms of progression-free survival. As long as that endpoint is agreeable for me, and that endpoint is positive, and the benefits outweigh the risks, I'm going to say, okay, you can go and market this. I don't have anything beyond that to do with what you are going to do with the medicines in the clinic. So that's what the FDA's mandate is. Now, this has to be clearly understood because we kind of put the blame on the regulator for everything that we don't like about the drugs and the way we practice or use those drugs, which is not really appropriate. But there are many other things that we will go through during the course of this talk, which will critically analyze those issues. Now, one of the things that we have been discussing very frequently today or in the recent past is accelerated approvals. Now, accelerated approval started in 1992 during the HIV pandemic or epidemic because you needed drugs really fast to come into the market. And so this is how drugs can qualify for accelerated approval. Drugs that treat a serious condition, cancer is serious, provides a meaningful advantage over existing therapies when the data is submitted to the FDA based on the small study that they are looking at, with a surrogate or an alternate endpoint that is reasonably likely to predict mortality. The reason why I have highlighted meaningful and reasonably is it can be very subjective. There's nothing very objective about being reasonable and being able to say that it's meaningful or not. But what is more important is that there should be a confirmatory trial ASAP. Now ASAP can be again very subjective, and the design has to be agreed upon. Okay, so this is how fast track, this is how accelerated approvals are done. So there's a lot of subjectivity when it comes to accelerated approval, except the fact that cancer is a serious condition, everything else can be subjective. Now remember again that this is different from fast track, breakthrough designation, priority review. This is something like Tatkal. Okay, the other things. So you can rush through, you can keep dynamically submitting data, you can go to the FDA repeatedly and tell them this is the data I have, what else do you think you will want to fast track my drug, and so on and so forth. But that is very, very different from accelerated approvals. Now it goes without saying that the maximum number of accelerated approvals have happened in oncology, and in the last decade, if you call 2013 to 2022 as the last decade, it's 123. In the last 5 years, every 3 out of 4 drugs that were given accelerated approvals were oncology drugs. So every 3 out of 4, that's a big number. But I think what's important is in the lower right corner of your slide. What have we done and how have we done this? Verified clinical benefit has happened only in half of all these accelerated approvals. Ongoing is about 1 in 3 drugs, 12% withdrawn. But the other thing I want you to look at is the median time for accelerated approval to full approval that can range anywhere from 6 months to 17 and a half years. Okay, 6 months to 17 and a half years. The other highlight that I have done is that box there. Time from accelerated approval to withdrawal can be as long as 4 years. Range is 1.3 to 12 and a half years. So can you believe that you've been using a drug with unconfirmed benefit for as long as 12 and a half years?
The company has made billions of dollars.
We'll come to that in the next slide. Right? Company has made billions of dollars, but it can dent so many other issues, which is what I will show you in the next slide. And even worse is the one on the left corner of your slide. In 2021, 10 negative confirmatory trials for accelerated approvals were done. 4 drugs were withdrawn by sponsors, but I think the worst part is the ODAC, said that you can still continue 4 out of the 6 things that the sponsors didn't withdraw. So you had a negative study, but the ODAC decided to say, no, it's okay, continue to market these drugs. And even worse is that these are not reflected in a lot of our guidelines. Okay? The guidelines clearly don't tell you whether the drug came into the market with full approval or accelerated approval, and how far are we in terms of a full approval, and so on and so forth. And the reasons that the ODAC gives is that the control arm performed better, so the test arm looked like it was not good. The test arm numbers seem to be as good as what was in the trial that led to the accelerated approval. So you can justify everything that you want to do and justify, but it doesn't look something that we will accept as a justification. So what does this lead to? It has impact on the sponsors, regulators, physicians, and patients. Okay, for sponsors, it tells you that the FDA is a very lenient body. You don't have to do a good proper trial with a robust endpoint. Just go and keep on bombarding them with accelerated approvals. You can make your money. Okay, so that's what it tells the sponsors. For the regulator, I think their belief and the trust that all of us put on a regulator is kind of dented, right? We say FDA has approved, EMA has approved, go ahead and use the drug, but this is what it can be. And it's even worse when there is a negative trial and the ODAC says, no problem, continue to market. Even when the sponsor wants to withdraw it, the ODAC has said, it's okay. If you want to keep it, you can still keep it. For physicians, as I said earlier, we just don't know most of the time what kind of approval that the drug has had, right? We just talk about it and then we forget about it. It's there in the market, so you go ahead and use it. So there's a lot of wasteful expenditure, as you rightly pointed out, especially if the confirmatory studies are negative. And most importantly for our patients, they are on potentially toxic, ineffective treatments. And most importantly, from an ethical standpoint, I think the regulator and the physician are betraying the patient's trust and sacrifice, especially those who go into that clinical trial itself, right? So I think there's a lot to think about. So the FDA has thought about it. So it's not that they're apathetic to what's happening. So this is what has happened last year. So there are some key differences, that have been put into this accelerated approval story. So it prefers that you should do a randomized trial, and this randomized trial should be ongoing at the time of accelerated approval. Because there's data to show that if you don't have an ongoing study at the time of your accelerated approval, you're very unlikely to do that and get back to the FDA with data. So this is a mandate, and it emphasizes that there should be a comprehensive plan that is put in early to verify clinical benefit. So it's better, but it's still far from optimal. So next time when you see an accelerated approval, keep track of what is happening, because it could end up being a negative study. And the third are ongoing confirmatory studies. It's only half of them which actually end up showing benefit in a confirmatory study. But then all of us should also take a step back and ask, should you always ask for a confirmatory study? Right? Because all of us will say, no, I want phase 3 randomized study to show that one drug is superior to the other with some acceptable endpoint. So the classic example is that of selpercatinib. 84% response rate, median PFS in the first line is 22 months, CNS overall response rate 85. Super. The FDA said, go ahead, get into the market. All that I want is the phase 4 data. But the EMA said you have to do a confirmatory study. So what happened? The libretto was done. And the libretto showed you exactly the same numbers as what the phase 2 study showed. But look at what happened. In the control arm, which is chemotherapy, investigators were allowed to use immunotherapy. We all know immunotherapy does not help in RET fusion cancers, but 80% of investigators decided to use immunotherapy. If you want to enroll patients who have a 1.7% incidence of RET fusion, imagine the number of patients that you have to screen before you did this study. And most importantly, you have that data. How are you going to convince your patient that you should take chemotherapy in the first line when chemo gives you a 65 response rate, which is still better than the run-of-the-mill pemetrexed carboplatin, and gives you 11-month PFS, which is still better than in many other situations. But you know the data, you have the data, but you're mandated to do a confirmatory study. And in a way, it is probably not ethical to do a confirmatory study in a situation like this. It's something for you to think about. I am not saying that you should not have confirmatory studies, but I think we should be reasonable as to where confirmatory studies have to be there and where it need not be, because who's bearing the cost of all of this? Ultimately, our patients. So if you're saying, I want to cut down the cost, then the cost of drug development also should come down. So that much about what the FDA has done and not. Now let's get back to the way we are doing practice. Let me ask a question here. These are two Kaplan-Meier projections that I'm showing. This is biliary tract cancer, TOPAZ and KN966. How many of you have explained the numbers on this Kaplan-Meier curve to your patients? Please raise your hands. Lovely. Amish, what did you tell your patient, Amish? No, I just want you to tell, there's nothing right or wrong here. I just want you to tell me what you say.
So I give them the realistic picture.
What's that?
Realistic picture is, if you use durvalumab. Well, I've tried to explain today morning in the breakfast what we were discussing.
I just want to know what you
One of the thing which I tell them, and in the durva arm specifically at the end of 2 years, what is the difference between the control arm and the durva arm?
Okay, so you will say there's a 10% difference.
Correct.
Correct. Sorry, Sahoo.
For
this, I specifically tell the GB is not doing well. I will give it because of.
Okay,
the thing.
so we are not
So that-
contesting whether you should give or not. I'm just trying to
so
understand
I
how
speak
you
to them
will
about
counsel.
this part of it.
Okay, lovely. Lovely. Okay, so that's a statistician's way of looking at it. So, Ullas is, so let's quickly run through these. These are two studies done in biliary tract cancers. The median overall survival is what we generally tell patients. You know, patients will ask you, how much more will I live if I use this intervention? Unfortunately, it's 6 weeks. Okay. Remember erlotinib, gemcitabine in pancreatic cancer, all of us laughed at it. Today it's immunotherapy. We are kind of dissecting through this data and giving it a second thought. And we'll talk about the tail of the curve. Okay, this tail is an unreliable tail because the number of patients at risk is single digits. So we are talking about this because there's nothing else to talk about in this. Not because we think that's the best thing to tell our patients. Okay? For us, it's a big jump in terms of what we have achieved in this cancer because in 50 years, we have not achieved anything at all. But that's a big achievement for you. What does it mean to your patient? I think that's what you should be thinking about more often rather than saying it's a scientific advance which makes you live longer for 6 weeks. Okay? So what are the numbers here? Actually, this is the number that all of us should be talking about because a Kaplan-Meier is meant to tell you what the hazard ratio is. Okay? This is something that we talk about, but we most of the time talk about this tail of the curve at a time when the tail itself is an unreliable tail because the number of patients at risk has come down to single digits, and we talk about it because that's how it's been presented even in the best conferences. In fact, I'm not showing you a terrible slide of the Topaz where they would have split this hazard ratio into two halves. You do a hazard ratio exactly to not do that. It is to tell your patient that wherever you are on the curve, this is the risk reduction that you're going to get. And in, I think, ESMO or ASCO, I don't remember where, on a podium, they did this. Wherever it is. So I think, and then for me, statistical significance is not equal to clinical significance. This is a statistically significant result, but the clinical significance is questionable. And why I'm saying questionable, we'll come to that in a little bit. We all, yeah? Yeah, so we'll come to that in a moment. So the p-values can help sometimes confirm the robustness of a negative result. If you're getting a good p-value and it's reproducibly bad, it reinforces that this strategy is not a great strategy for this disease. And for me, it should be a winner irrespective of which number you're looking at, whether it's hazard ratio, whether it is analysis of numbers at a particular point, or the median. TDXD is a classic example of that, you know. Nice separation early, the hazard ratio is so good, and you know, whichever time point you're doing that analysis, it's got a big difference in terms of outcome. That is a good intervention. That helps most patients. This is not something that is going to help most patients, and we've already seen that in our practice. There's always an odd patient who will do well, and that's the patient we always remember. We don't remember most of our patients who don't do well on a particular intervention. We come to the question of surrogate endpoints, right? This is something that keeps bugging us all the time. So this is the Pembro trial in kidney cancer, okay? This is the first reporting that was done in 2021. Now, the hazard ratio here is 0.68. The 24-month difference is 9.2%. It's not some 1%, 2%, it's 9.2%. So let's take a step back and ask, who decided that this is a good surrogate endpoint? It's people like us sitting somewhere else in the world, but it was agreed by a group of people. It was not put into place by one person. I think the questions that we have to ask ourselves and ask our patients is living with disease okay for you? Or living without disease? Because we can always tell them, I've got this intervention that you can use later, but you will live with disease, you'll have to be treated for an endless period of time. It comes with a lot of toxicities, and of course the cost can be more. Is that acceptable to you or not? I don't know how many of you have looked at the kidney data updated overall survival. The hazard ratios for overall survival are terrible. It's 0.79, 0.8. Okay? Not something that all of us would love to see. All of us, at least I thought it will be a super hazard ratio that you will end up with in the overall survival. In fact, if you look at the Len-Pembro data, the Len-Pembro does 1 month poorer compared to the test arm. I don't know how many of you noticed it. If you have not noticed it, go back and look at it. So we just don't know how good that overall survival benefit is going to be. I think what is important for us to look at is how good is this PFS benefit? How good is this DFS benefit? And I'll tell you my perception of what is good and what is bad. Of course, you can have your own, but how good is this PFS benefit is important. Are we causing some detriment in terms of overall survival? Because they will show you a projection at that time. Is this okay or not? This is not trending in the wrong direction. So for me, if the magnitude of benefit is decent, and as I said, I'll tell you quickly what my definition of decent is, and if the overall survival is trending in the right direction, then I'm okay to use this surrogate endpoint to give this to my patient at this point in time. If it's not, then I'm not going to do it. The other question we'll have to ask is, this is the result for today. If this is what it is 8 years from now, am I okay or will I kick myself? Right? If I answer that as an affirmative, I'm good with what I'm seeing today. I'm not going to tell myself, why the hell did you do it if the overall survival does not come after 8 years? I'm still okay. Go ahead and use this. And immediately the next question comes as, if this happens, will you kick yourself for not using the intervention? Because this has ended up in an overall survival. So from 2021 to 2024, if you had denied somebody Pembro, are you going to tell yourself that I did something that I should not have done? Can you justify it for yourself and your patient is a question that you should always ask. And 70% of all patients here had IO later lines. Okay? It's not Pembro, but they had some IO in the later line. So these are some things I think we have to think about. Amol, I'll give you a chance to talk. Don't worry. Okay, we'll have some discussion after that. Though I'm going to overshoot my time, but then I think these are things that we have to think about. So this is my definition of a small, a large, and a good benefit. Right? If your hazard ratio is greater than 0.8, 0.8, 0.9, and so on, that's bad. If it's 0.6 to 0.8, it's good. If it's less than 0.6, it's very good. If it's less than 5% absolute difference, it's not a great intervention. If it's between 5 and 10, it's good. But if it's more than 10, it's really good. And that's how I keep this in my mind when I'm talking to my patients. And this is what I tell them. It's good, bad, ugly, or whatever it is. But you push these numbers up and down. It's very subjective. This is not what it is. So I will, So sometimes the Patiala can be bad, you see. The next day you will realize it. So it's not all the time. Okay, so this is about subset analysis. So again, I took something that was recently discussed in our WhatsApp group, and yesterday, Sahoo discussed about it. So FDA approves Pembro with chemoradiation for 2014 stage 3 to 4A cervical cancer. So the criticism was, why was this group just left behind? Right, that's one. And then yesterday we didn't discuss about PDL1, so that was discussed in our WhatsApp group. So, the approval should have been all patients who went into the study, who have a PDL1 that's positive, and who are taking IMRT. Okay, so to do IMRT or not is not the FDA's business. So we leave that aside. Okay. Now, remember that this is 45% of all patients. Now, I'm just trying to step into somebody in the FDA shoes and say why this may be justified at this point in time. Of course, you can say that that's not valid, but I'm just trying to, justify what was done. Now, all of us spoke about short follow-up yesterday. Everybody said, I won't use it now because the follow-up is very short. Absolutely fine. So here is a group of patients who don't seem to be deriving benefit, at least in the short run. You're using an expensive intervention. It's a toxic intervention. So I'm not too sure whether at this point in time we should push it to everybody who will fit in the clinical trial criteria. It's okay to wait before we say everybody can take it. Let's wait for a longer follow-up. Let the hazard ratio for this group get a little better before I say, okay, go and give it to everybody. Because there is some toxicity with immune checkpoint inhibitor. We cannot say it is like water. Why not PDL1? Sir, can I take 5 minutes? Okay. Thanks for that. So, lovely. So, why not say only PDL1? Now remember that in cervical cancer, almost everybody is PD-L1 positive. Okay, it's 4% of patients who are PD-L1 negative, and we know the problems with PD-L1. So for the sake of that 4 patients, for the sake of this, the other 95 who are going to be PD-L1 positive, can you throw away the 4% and then say, no, you are not eligible for the drug? I don't think that's fair, right? Because you can say that in the advanced setting, only patients who had PD-L1 benefited, but 10% were PD-L1 negative. So when you look at subset analysis, I think you should be very careful to the way you interpret it. So for me, there are ways and means of justifying what has been done by the FDA. We may have our opinions, that's okay, all of us are entitled to our opinions, but is this justified? For me, the answer is yes. And remember that you need to have pre-planned and powered. There are pre-planned subset analysis which are not powered. Please remember this. These are 2 different issues altogether. And multiplicity correction, which, in the previous, journal we all discussed. And I think if they're not reporting something, look at the consistency of this. L858R, every damn study that you look at, there's a problem there. So it is very consistent. Even if I'm not reporting a p-value, I know that there's something that's happening there which is making your drug not work as well as what it is in exon 19 deletion. So I don't want a p-value there. It's consistent. Can you explain your subgroup analysis with a biological rationale? We know that L858R is associated more often with P53. It's associated more often with other mutations. So yes, we know that there is a clear biological rationale behind why this group of patients don't do well with the existing interventions. Okay, so look for all of this in a subset analysis. Don't go and just look at the confidence intervals and jump saying it's exceeded 1, so it's useless. That is a very elementary way of interpreting a subset. And remember that in 9.5 out of 10 times, a subset or a subgroup analysis is only hypothesis generating. Nothing beyond that. We don't have anything else to do, so we just go and keep on torturing that subset analysis and trying to get all the nonsense out of it. It is not meant to be that. Please remember this. For want of time, I'm not going to this. Suboptimal control arms in oncology. So this is another lovely analysis that was done. So about 1 in 5 studies have a suboptimal control, and the top are GU and hematology. Okay. Now, how can you justify this?
Accident or by design?
By design. These things don't happen by accident because so many people are looking at it, right? So what is the justification? The justification first is there is no standard of care, and then it is a multi-country study, and what is available as standard of care here is not available there. As always, you can justify everything. But what's the problem there? Whenever you do a trial like this with a suboptimal control arm, your events accrue very fast. You can actually complete a trial very, very fast because the events will happen, because the other arm is just coming in rapidly with events, and your test arm, even if it is not so great, is going to go slowly in terms of the events. And remember that all of these are event-driven trials. So the faster the events happen, the faster you will get a positive study, the faster you can get into the market. But it does not answer this question. So this is something that should not happen. So how can we overcome this? But this is something I call as the uro-oncology paradox. On one side, you find that most of these bad trials are in uro-oncology, but some of the best studies are in uro-oncology, right? So look at the PEACE study, super design. We discussed about it yesterday. They kept on changing I think it is just brilliant, when I look at the way things would have evolved over time. One after the other, you keep on changing the standard of care arm, make sure that you're calculating the numbers that are necessary to give you a robust result. Super. Stampede is again a brilliant way of doing studies. You keep on building up over your previous, control arms, and then you can compare your new arm to your previous control, compare the next arm to this. I think it's just a brilliant way of doing studies, and then this is always there. Dealer's choice of treatment. If you don't have a standard, let people choose. Okay, at least you're not blaming that it is suboptimal. We are the guys who chose the good ones, the bad ones, or the ugly ones. We have to take responsibility for what's happening. Some of these approvals became irrelevant by the time they come to the market, and these are 3 approvals which are already irrelevant. Okay, this is a combination of PARP inhibitor and an ARPI in patients who have had docetaxel and ADT for their HSPC. This is CRPC. Tell me how many of you are not using combination therapies in HSPC now? Yesterday, we had a discussion about docetaxel plus ADT. Please, ladies and gentlemen, yes, there are a couple of ladies here. ADT plus docetaxel is not routinely used anymore in HSPC. Okay, we can, as medical oncologists, don't want to let go of the docetaxel saying, if it's visceral disease, I want to use visceral disease. Tell me a good data set that tells you that docetaxel is better than an ARPI in visceral disease. There is no data. There is some torturing of data and subset analysis to show that in liver metastasis, docetaxel may be better. So we also want to adopt subset analysis that are unscientific to our convenience, right? So everybody in the HSPC today would have had ADT plus an ARPI. And if you had an ADT plus an ARPI, you're not eligible for any of these. Okay? Already in the bucket by the time they reach the market. So that's something that we need to keep a watch on. And again, Ullas wanted, when we discussed in our group, what do you think should I speak about? He said value. Remember that you cannot put a price on life. Okay, every single day you live is important and the cost of drugs are going up. It's not as simple as this. I think we need to decide on value based on whether you're talking about value for the system when somebody else is paying or when you're paying from your pocket. And I also think it depends on these 4 Ds. Disease. How rare? Is it early disease versus metastatic disease? Because your threshold to accept the values or the numbers in that study will vary very much based on this. The drug, how efficacious or toxic is it? The dollars or the cost and your desperation, right? Let's imagine a 25-year-old guy with some rare malignancy who has a rare treatment which is not so very beneficial vis-a-vis an 85-year-old with the same disease and coming to you. The 85-year-old would gracefully accept, okay, doctor, it's all right, I'm fine to be left alone. The 25-year-old, even you are not prepared to leave him alone. So the value will change based on multiple factors. You cannot quantify and say, this is valuable, this is, even the ESMO and the ASCO don't agree on this. Please remember this. So there's no way that you can say, this is valuable and not. What magnitude of benefit, p-value and toxicity are you prepared to use a drug has to be decided by the individual. If Ayushman Bharat is going to fund for you, the issue is very different. But if you are paying from your pocket, the issue is very different. And this is what patients think. 1 in every 4 patients wants to take an intervention as long as your overall survival is better by 1 month. Okay? 1 in 4 will take an intervention if your overall survival is better by 1 month. But the same blue bar here is 1% of physicians. So there is a physician-patient disconnect when it comes to what value is and what they are prepared to take for what benefit they will accept. Please remember this. It's very, very important to tell your patient. That's why I said I will justify use of the drug in biliary tract cancer. This is exactly the reason. If your patient thinks living 6 weeks on an average extra makes sense to him or her, by all means, you should use this. But the point is, you should be fair in having that discussion with your patient. And again, I want to stress on this. We want patients to live longer. We want patients to live better. But how long, how better has to be a rating and weighting that is to be done by the patient, not necessarily by regulatory authorities and physicians. But do we have that? Are we going to get there? The answer is probably a no. I'll finish in a minute or so. So how you view things matters a lot. Remember that when you're doing any analysis, it's to do a critical analysis, not to find fault. There's also this notion that anything that's expensive is bad. And it's seen with the magnifying lens where anything that is simple and cheap, you will say it's cheap, that intervention is fine, let's go ahead with it. I think that's very unfair. And ladies and gentlemen, remember that the FDA consists of people who have seen much more of the world and the drugs and all of this than you and me sitting in this room. It's easy to criticize. Remember that it's decided by vote. It's not one person who is going to decide. The ODAC will vote and the FDA will take a call on that vote. And trust the regulator, but use your discretion. You have a brain, right? Why do you have to blindly follow what somebody is saying? And remember that there's nothing called as perfect, and even if you're perfect, you're not going to succeed all the time. Okay? So keep this at the back of your mind. I just want to show you two instances where there is no absolute good evidence, but all of us follow it. How many of you use capecitabine for biliary tract cancer adjuvant? How many of you gemcitabine carboplatin in the same setting? For both, there is no evidence. So, Amish, you can be happy if you're using neither, right? So, this is BILCAP. Okay, this is what all of us use. Tell me where in this slide you can find a statistically significant, clinically beneficial result. The answer is no. Some of us use gemcitabine and platinum. I have used it sometimes. Consistently negative results for gemcitabine and platinum, but all of us would use it, right? So, we do things to our convenience many times. Folfirinox, okay? I just read through these numbers, okay? This is chemotherapy, so nobody thinks about it, nobody minds it. We all want to discuss only immune checkpoint inhibitors. All cycles of chemotherapy, only 2 out of 3 got it. Early stoppage was 1 in 3 patients. Nobody did a censoring and analysis and, informative censoring for this trial. Look at the difference. So it's about 10% difference if you do an informative censoring, unequal informative censoring. This is overall survival benefit. So if you went ahead and did an informative censoring, and I'm not a strategist, I don't know how to construct those curves, you will see that the benefit dwindles down to much lower than what you get with Prodigy and Folfirinox. How many of us went and did this? We didn't do this at all. We blindly say anybody who's got a periampullary pancreatic cancer should go on Folfirinox because that's what improves your overall survival. Look at this. A breast cancer oncologist will spit at this dose intensity. 48.7%. Okay? All of us want to do 85% plus. As students, we are taught that if you come down to less than 85, gone. But how will you justify this? In spite of only 48.7% getting a more than 70% dose intensity, the trial still was positive. I don't think that's a great justification of using Folfirinox for these set of patients. So think about it. Some patients, some drugs and subsettings seem to be privileged. 3 drugs in breast cancer, pertuzumab, Keynote 522 and abemaciclib. All of them disease-free survival. No overall survival. All of them almost have the same numbers. We just, kill abemaciclib and we almost kill pertuzumab, but nobody talks about Keynote 522, right? So some drugs, some diseases, and some patients seem to be very privileged, and I still don't know why. So guys, Amol, I haven't completed yet. So please don't think I'm talking. Okay, guys. So I'm almost done. So self-regulation, I think, is the best regulator. Okay? So you need a regulator. You need the police. You need the politician. You need the bureaucrat. We may have our complaints against all of them, but remember that if they are not there, you will be a rogue nation. Right? Whatever it is, Tom Cruise or whoever it is. So that's why I said, you can justify anything. All is fair in love and war. And now I've added oncology to it. Right? You can justify anything that you want to do. Please do. Because as long as your patient is fine and you're fine with it, it's okay. And this is why we talk about evidence-based medicine. Clinical research is insufficient. In fact, a lot of situations where evidence does not exist. And that's why you are called an expert. You have to trade the risks, benefits, patients' values, guidelines, so on and so forth. And that's why we all say medicine is not just a science, it's an art. And that's extremely important. And that's why at the end of the day, if you treat a disease, you win, you lose. But you treat a person, that you will always win no matter what the outcome is. And I'll stop there, because I've taken a lot of time, and I'm happy to take any questions, criticisms, anything that you want me to. Thank you.
Can I have some comments? If I'm allowed, with the duration of time.
I'm going to turn this way.
I have 4 comments based upon the slides that you have shown.
Oh, yeah, sure, Amol. I'm happy to answer.
First one is you have shown about the adjuvant RCC pembrolizumab data, and you have shown that there is an OS benefit now. But what we need to understand is that whatever that is published, of course, need not be taken as correct because it is the same trial for the same curves. Ian Tannock and group has published curves with removal of the censored data, and it is published in Lancet. Not
I've gone
in
through
some
that.
ordinary
Amol. I
journal.
know you will be here and I've gone through it.
And they have said that there is probably no benefit. So we cannot just assume that because it is published and it is there, and in that publication, they have not given the numbers of the censored patient. They had that entire good practice of showing in the bracket is gone. So there is no transparency in the data. The data can be accepted provided there is transparency, and it has been shown in that particular incidence.
So can I answer that, or you want to finish all the four?
Yeah.
You will finish all the four, lovely.
You answer one at a time.
Okay.
Okay, so I think that was a very early analysis of the curves, right? So it was when the 2021 paper was published. So now it's 2024, right? So the data that you had was at that point in time. So there are more events that have accrued now. So based on the more events that have accrued now, we are now showing an overall survival benefit. As I said, you may have your problems. So informative censoring happens in every study. So let's go back to Monarch, because that's where this informative censoring became a big issue. Remember that one of the complaints that we had about Monarchy was that it's overpowered, right? Yes. 5,000 patients. Now let me ask, the people sitting here. So I went and looked at the numbers that Ian Tannock and group, with all respect to, the group that has written the criticism. So the difference in terms of overall survival events in between the two groups, right? The censored group is 10 patients. So we are saying that you're overpowering a study, but then we are saying that a difference of 10% or 10 patients in a 5,000 patient study can make the results negative. Now, I'm not a statistician again, but I just want to put some logic there. How can, in a 5,000 plus patient study, make a difference based on 10 patients? It is unlikely. It is unlikely. So let's not, I don't want to discuss the statistics, Amol. So I think we have to take both the sides of the story, right? There will always be informative censoring in every study. What about the other things that I showed? So it is going to be there. So we need to use our discretion at the end of the day and then say, okay, these are the numbers that we are seeing. And informative censoring, how many of you who have used immune checkpoint inhibitors think that 18% will be discontinuing therapy because of toxicity? Please put up your hands. 18%, that is the discontinuation rate in Keynote 564.
1.8, not 80, 1.8.
1.8, yeah, 1.8. So in Monarchy, 18% discontinue. So that's, if students are here, if your Ullas asks you how many patients discontinue, 18. In most studies, 18 is the discontinuation percentage. Okay? So I think we have to clearly see the fact that in real life, there are ways and means of working around toxicity, but in clinical trial, it's not there. It's hard and fast. If your patient has this once, you're out and you don't come in. So I'm not disregarding anything that Dr. Tannock and group have said, but I think it's a little overstating the facts rather than the right perspective.
So let me pick up your words that you said that there has to be transparency and both sides should be shown. At least they should show the censored data.
Only Lancet Oncology does that.
The rest of it doesn't do.
Do you want to continue with your next?
Yeah. No, next question. Now, I had 4 points. And
please.
No, you said about in cervical cancer, PDL1 less than 1% was only 4%. 4%, less than 1%, 4%. It doesn't matter in cervix, I agree. Probably they could have not done that subset analysis only when everybody knows.
They didn't. They didn't do it.
No, it is shown in their own publication. They have shown that data, of course.
No, that's in the advanced setting, not this setting.
So in this particular trial also, that figures are there. That is how everybody knows that those figures are there. They're published. So
no, this study is not published yet.
Whatever the literature that has come, the presentations and all.
Okay.
So I will give another example how these particular things are covered up. Suppose we have an esophageal cancer, an adjuvant nivolumab, the benefit is more than CPS more than 10. More than 10 can be more than 10 to 20, 20 to 30, 30 to 40, 40 to 50.
I agree with that point.
What about that particular subset?
I agree with that
never
point.
given.
I agree with that point.
It is possible that the benefit is only above 40 and 10 to 40 has probably no benefit. So this is how they should give more information. They should be more transparent
so I think we
so
should
that there is more belief on their data.
I entirely agree with that point. I don't have anything to say for that.
Okay, you agree. So you
Yes,
agree
I
one
agree.
point.
Yes, I agree.
Third point, you showed what the patient wants.
Yes.
You showed that things that yes, for patients, 1 month benefit is important. If you see the source of that information, that study has been done in a country where everything that patient wants is paid by insurance. If such similar studies is done in India, and most of it, we have 40% coverage, probably the results may not be the same. So in a system where patient is getting paid everything by the system, they will say, I want 1 month benefit because I'm not paying for my own
So
pocket.
that is why I qualified my statement by saying, is Ayushman Bharat paying or your pocket?
So
So
in
if your pocket is what you're paying, please, by all means, you have to, the patient is going to know that, in a month, this is what is the cost, and let the patient decide. That's the point I'm
and
making.
the last point about the pancreatic thing, you showed Folfirinox and
Yeah.
It is just last month only Vishal has published a paper saying that there is no progress has occurred in pancreatic cancer over last so many years. So there are people who accept that Folfirinox is not a real progress. It is just numbers which have been put up. So yes, people accept that whatever that is not good, there are people who say that it is not good.
But it's not as loud as what you speak about the many
Because
other
the problem
things.
is there
That's
is
the
no
point.
there is
That
no
is the
support
point. That's the
for
point.
that.
I agree, but it's not as loud as what we
So
would
you
like
agreed
it.
3 points.
Era. So the just Topaz, like my point is about the tail, and you spoke about the tail of IO. I have not only you, but objection to that particular statement. Topaz GB और biliary tract cancer हटा के non-small cell डाल दो in 2024, okay? And this non-small cell, all patients are given chemo plus IO, including EGFR, AL, cross, MET, all of them. And we will get a graph something like this only. So my point is that, and the number at risk, I think my understanding is slightly different from how you interpret it. Less the number of risk is more the robust data because the events have happened in those patients. In fact, more the number of risk in the Kaplan-Meier, the events have not happened and the data is not mature. I'll just complete my question. My point is, Senthil, like you showed that genomic alteration based treatment versus just IO upfront in all patients, 3.5 versus 2.6 months, what you have shown. I think biliary tract cancer, we have started understanding it now a little better with Topaz and Keynote trial. There will be HER2 positive trials which will be coming up in a positive way. I think each advance, even if it is tail, we have to take it in a positive way and move forward. That is what my suggestion is.
I entirely agree. I'm taking it everything positively. Otherwise, I wouldn't have said some of the good things that I said. Right?
So I don't have a question, I have a comment. Liked your FDA part of the presentation. Just a request to the colleague, be a little generous to your regulators. They have a really tough job to do.
Agree.
So, I mean, I
That's
do
exactly
sit on
what
a
I
few DCGI committees on drug approval.
I entirely agree. So that's
So
why
balancing
it's
everything
easy to
is,
sit here and point fingers.
It's easy to do that.
Anyway, thanks again for that opportunity.